Hi everyone. In this lecture, I will talk about mitochondria, biological oxidation, electron transport chain, and oxidative phosphorylation. The chemical bonds energy of lipids, carbohydrates, and amino acid must be converted into chemical bond energy ATP. This is not possible directly. Chemosmatic theory explains how we achieved all these processes. Theory established the relationship between the electron transport chain and ATP synthesis. Before starting this lecture, I would like to give a little bit information oxidation and reduction reactions. And oxidation reduction reactions is a type of chemical reactions that involves a transfer of electron between two species. And we have a lot of different type oxidation and reduction reactions in our cells during metabolic processes. When we look at these reactions, for example, loss and gain of electrons, in first reaction, we can see oxidation of zinc, and second one, reduction of cupric ion to copper, and also another oxidation and dehydrogenation reactions during carbon dioxide metabolism. We see oxidation of carbon and reduction of carbon dioxide by deoxygenation. Also, dehydrogenation and hydrogenation reactions, it can be occur in different type of reactions in our cells. For example, dehydrogenation of ethanol to acid aldehyde and hydrogenation of pyruvate to lactate. And in our lecture, we will talk about different type oxidation and reduction reactions. When we look at metals and oxidation reduction reactions, you can see zinc, copper, and iron atoms have this kind of ability. And also, when we check standard reduction potential of electron transport chain component, for example, NAD, NADPH, NADH, and NAD, FAD, FADH, also cytochrome B, A, and C, and cytochrome C has different type reduction potentials. And in this case, it can be explained why electrons goes from ferric atom to oxygens, because oxygen is most important reductants in our cell. And when we look at mitochondria, this mitochondria is a so unique organelle, and all reactions, electron transport chain and ATP synthesis reactions are involved in mitochondria, because when we look at mitochondrial metabolic processes, citric acid cycle and beta oxidation, also pyruvate dehydrogenase reaction, some amino acid metabolism reactions must be occur in mitochondrial matrix, because this is a big advantage for electron transport chain and ATP synthesis, especially during citric acid cycle and beta oxidation. We can produce NADH and FADH also we are able to produce NADH in cytosol, but this NADH producing in cytosol cannot be enter matrix. For this reason, when we produce NADH or FADH2 in matrix, these two molecules cannot go to outside of matrix and must be used in electron transport chain. And another important things for mitochondria, membrane structure. When we look at mitochondrial membrane structure, mitochondria has a double membrane. First is outer membrane, It is thicker than the inner membrane and similar to the structure of the plasma membrane. Also, outer membrane contains channel system and called porin. And there are different type enzyme associated with mitochondrial lipid synthesis in outer membrane. And other side, in mitochondrial inner membrane, mitochondrial inner membrane is a semi-permeable membrane type. Some molecules cross the matrix by the active transport. The inner membrane runs parallel to outer membrane, but it makes crystal folds towards the matrix because of its intense functional properties. Also, the protein ratio of inner membrane is higher than other membranes. And as I told you before, we have many important chemical reactions in matrix. Matrix are closed by inner membrane. Also, there are plenty of protein molecules, various reaction enzymes, including lipids and carbohydrate metabolism. Also, there is mitochondrial DNA in matrix, ribosome, and tRNA. The number of granules in the matrix is small. Excess of calcium and magnesium in the cell pass to mitochondria and bind to granules. This calcium and magnesium are returned to the cytosol when necessary. And another important issue for mitochondria, it is mitochondrial genome, mitochondrial DNA. Mitochondrial DNA encodes 12 proteins that are subunits of enzyme oxidative phosphorylation. Also, 74 polypeptides of the oxidative phosphorylation complex are encoded by the nuclear genome. In most multicellular organisms, like mitochondrial DNA, as a circular DNA and covalently closed, and it has double-stranded DNA. And human mitochondrial number is dependent on cell type and cell function or cell needs. And now organization of the electron transport chain. The inner membrane of the mitochondria has 5 separate or different type enzyme complexes, including electron transport chain and oxidative phosphorylation, NADH dehydrogenase complex 1, succinate dehydrogenase complex 2, cytochrome C reductase complex 3, and cytochrome C oxidase are components of electron transport chain, and F0 and F1 ATP synthase is responsible for ATP synthesis. And all protein complexes inside in inner membrane of mitochondria. And we have 5 type protein complexes during oxidative phosphorylation. And we know very well enzymes have coenzyme and cofactors for chemical reactions. Also, electron transport chain and ATP synthesis also have this kind of molecules, cofactors and coenzymes. For example, NADH flavoproteins FMN and FMNH2 and FAD, iron sulfur proteins, ubiquinone, cytochrome A, A3, B, C, and C1, and copper are found in electron transport chain component. And we know very well all chemical reactions related to these protein complexes, including these cofactors or coenzymes. And 3 types of electron transfer are observed in oxidative phosphorylation, and first is direct transfer of electrons. Also, is in this case ferric and copper atoms and ubiquinone are able to catalyze this kind of reactions. Another one is transfer as hydrogen atoms, FADH2 have this kind of reactions, and also NADH can be transferred hydrate ion during electron transport chain. And when we look at first complex 1 is named NADH dehydrogenase enzyme systems. Enzyme is very huge protein complexes, and it has 42 different polypeptides and at least 6 iron sulfur center and 1 FMN molecules. And also it is responsible for electron transfer and NADH electrons to ubiquinone. And the reaction occurs inside of these protein complexes with coenzyme and cofactor of complex 1. NADH gives 2 electrons FMNA, and FMN turn to FMNH reductor form, and second step is electrons in FMNH transfer or convey to ferric atom and iron sulfur center. The NADH when NADH give 2 electrons turn to NAD. This NAD must be go to beta oxidation or acetic acid cycle and convert to again NADH. It is another important circulation. And also FMNADH reductor form can be turned oxidative form because of next reactions, and iron sulfur center must be give electrons step by step coenzyme Q ubiquinone. And also electron transport chain complex 1 has another role and work as proton pumps. These electrons goes to NADH to ubiquinone. At the same time, its protons must be transferred from matrix to intracellular space in mitochondria. In complex 2, this a part of acetic acid cycle. It's a component of acetic acid cycle and binds to inner mitochondrial membrane. And enzyme use succinate as substrate and produce fumarate. Coenzyme of succinate dehydrogenase is FAD, and enzyme produce FADH2. And FADH2 electrons, 2 electrons must be moved to iron sulfur center, such as complex 1, and these electrons from iron sulfur center to ubiquinone. As this is another important reactions to related to iron and sulfur center. And all molecules, all electrons of NADH and FADH2 must be transfer of coenzyme Q ubiquinone. Ubiquinone has a different structure compared with complex 1 and complex 3. Also, it has one hydrophilic tail and accepted as in lipid forms. The hydrophobic nature of coenzyme Q keeps it localized in the inner membrane, mitochondrial membrane, where it floats around to transfer electron from complex 1 and 2 to complex 3, and we called mobile electron carrier. When you look at its function of ubiquinone, ubiquinone oxidative form is called ubiquinone, and ubiquinone accept one electron and turn to coenzyme Q semiquinone intermediate, a kind of radicals. These molecules, semiquinone intermediate of ubiquinone molecules, is one of the dangerous free radicals in the cells, because these molecules, electrons, can be go to mitochondrial DNA and membrane of mitochondria, or leakage to matrix and affect to cellular cell membrane or nuclear DNA. And for this reason, this coenzyme semiquinone and received a second electron and turned to coenzyme Q reduced form, also another oxidation and reduction reactions in the cell and mitochondrial membrane. And also these electrons must be go to other complexes, complex 3. Also, we know very well, and there are a lot of disease related to ubiquinone and coenzyme Q dysfunction, because it's a key molecules transfer of electrons, NADH and FAD electrons to complex 3. And we know that very well we can produce NADH in aerobic glycolysis, and during this condition we produce 2 NADH molecule, but NADH in cytosol cannot pass to other mitochondrial membrane, and when we need electron of this NADH, we have to transfer these electrons inside of intermembrane space or matrix. And also we have two type system, two shunt system for transfer of electron of NADH. First is called glycerol triphosphate shuttle. We have two type glycerol triphosphate dehydrogenase enzyme, cytosolic and intermembrane space type. Cytosolic glycerol triphosphate use dehydroxyacetone phosphate and substrate turn to glycerol triphosphate, and dehydroxyacetone phosphate receive 2 electrons of NADH and turn to regular form glycerol triphosphate, and glycerol triphosphate is able to pass other mitochondrial membrane and enter intermembrane space, and that places glycerol triphosphate turn to the dehydroxyacetone phosphate again by glycerol triphosphate dehydrogenase intramembrane space form. Enzyme is name, but also chemical reactions same, but it coenzyme is different. First enzyme use NADH, but second one, intermembrane space dehydroxyacetone phosphate, use FAD as a coenzyme substrate and produce FADH2. These FADH2 electrons also move to ubiquinone, and in this case we should have transferred NADH electrons starting or enter electron transport chain in complex 1, but in this case these electrons enter the electron transport chain directly ubiquinone system, and it can be some problems for ATP synthesis in this case, because we cannot use this first enzyme system complex 1, and we cannot pass protons from matrix to intramembrane spaces. And another complex name called malate aspartate shuttle, and also this enzyme has two types, intramembrane cytosolic form and mitochondrial form. And cytosolic form enzyme, dehydrogenase enzyme, use oxaloacetate and turn to malate, and also coenzyme is NADH, and malate can be entered by malate alpha ketoglutarate transporter system, and malate enter matrix and turn to again oxaloacetate, and enzyme malate aspartate dehydrogenase matrix form, mitochondrial form, with NADH coenzyme and produce NADH, and we are able to NADH electrons in cytosol can transfer these electrons into matrix. It's a good advantage for ATP synthesis. And complex 3 and called coenzyme Q cytochrome C reductase. This cytochrome B and C1 complex called complex 3 and spans in inner mitochondrial membrane and translocates protons to intermembrane space, where is able to also electron transport from ubiquinone to complex 4. Complex 3 contains 3 different iron containing cytochrome proteins, cytochrome B and C, and also another iron sulfur proteins called Rieske protein. Also, the iron in the cytochrome proteins is located in Phosphorilation ring system structure, and electron transfer is primarily carried out by cytochrome B, then to cytochrome C1 and the Ham iron sulfur center, and cytochrome C1 interacts directly with the mobile electron carried cytochrome C, another type of cytochrome. And cytochrome C is mobile type of cytochrome, and located in inner membrane, intermembrane spaces side. And the role of cytochrome C is to carry electrons from one complex of internal membrane proteins of inner mitochondrial membrane to another. And the electron donor is cytochrome C1, as I told you. The electron acceptor is cytochrome C oxidase, and electrons cytochrome C1 to cytochrome oxidase can transferred by cytochrome C. Both these integral membrane proteins have binding site of the peripheral membrane protein cytochrome C. Cytochrome C enables transfer of the electron by force binding to cytochrome C1 and subsequently binding to cytochrome C oxidase. It has a key role between cytochrome C1 and cytochrome oxidase. And when we look at last complex of electron transport chain, named called cytochrome oxidase. It is the last enzyme in the restore electron transport chain of cells and located in the membrane, inner membrane. It receives an electron from each of 4 cytochrome C molecules and transfer this electron to one dioxygen molecule and converting the molecule of oxygen to 2 molecules of water. And when we look at structure of cytochrome oxidase, the complex contains 2 hams and 1 A cytochrome and also 1 cytochrome A3 and 2 copper center, copper N and B centers, and cytochrome A3 and copper B form a nuclear center that is site of oxygen reduction. And cytochrome C gives electron copper A, and electrons move to cytochrome A, then copper B, and copper B transfer electron directly to oxygen and produced 2 molecules of water. Of course, we don't have an aim to produce water in electron transport chain, and we want to produce ATP during all these processes. And we have one question: if we transfer electrons of NADH of electrons of FADH2 in the electron transfer chain, and in this case complex 1 and complex 3 and 4 have another ability and work like proton pumps, and is it enough to synthesize ATP? No, we have to transfer electrons NADH and FADH to oxygen, but it's not enough to synthesis of ATP. And we have to explain ATP synthesis or oxidative phosphorylation, and we have one theory or one model and called Chemosmatic Model. The Chemosmatic hypothesis or theory was proposed by Peter Michel, and this hypothesis based on a proton motive force was responsible for driving synthesis of ATP. We have to connect or electron transport chain and ATP synthesis, and this theory explains how it is possible. In this hypothesis, proton would be pumped across the inner membrane, and as electrons went through the electron transfer chain, and after that all chemical reactions, all electron transfer chain reactions result in a proton gradient with an lower pH in the intermembrane spaces, pH degree decrease, and elevated pH in the matrix of mitochondria, a little bit is basic compared with intermembrane spaces. And another activity of these electron transport chain reactions, and we produced a proton gradient between intermembrane spaces and matrix. Also, we can produce a voltage gradient between intramembrane spaces and matrix. It is important. It is most important things and explanation for ATP synthesis. The proton gradient and membrane potential are the proton motive force that is used to drive ATP synthesis, and in effect the pH gradient acts as a battery which stores energy to produce ATP. And this chemosmatic theory has been accepted as the mechanism of coupling of electron transport and ATP synthesis. And after that, Peter Michel received Nobel Prizes in chemistry 1978. Now we have an important aim during all process of oxidative phosphorylation. We have to synthesize ATP. This ATP referred as a molecular currency for intercellular energy transfer is adenosine triphosphate. ATP is a nucleotide form of triphosphate form of adenosine nucleotide, function as a chemical fuel by powering many organic processes of life. And ATP generation is the principal energy generation procedure found in all forms of life. And ATP is the fuel for the operation of almost all metabolic pathway of cell. In an ATP molecule, two high energy phosphate bonds called phosphoanhydrate bonds, which are responsible for high energy content. Hydrolysis of the third phosphate group produces adenosine diphosphate, an anorganic phosphate. Along with a considerable release of energy, ADP can absorb energy and regain the group of regeneration and ATP molecule to maintain constant ATP concentration. And we know very well is ADP-ATP balance, also NADH and NAD balance are so important for regulation of electron transport chain and oxidative phosphorylation and ATP synthesis. And when I want to look at ATP synthase enzyme structure, which is responsible for ATP production. An ATP synthase enzyme provides a channel for those protons, and it has a key role of ATP synthesis. And the potential energy from these gradients, proton gradients, is often used to perform biological works. And ATP synthase, also called F0, F1 ATPase, is a rotary motor enzyme. The enzyme is found in the membrane mitochondria, and the enzyme consists of two parts: the membrane-bound F0, which is a proton translocator, and F1 part, which has catalytic activity. F0 part can be considered to be a rotary electric motor powered by proton flow, proton motive force, while the F1 part acts as a rotatory chemical motor powered by ATP hydrolysis. The enzyme is reversible. As if you can separate F0 and F1, F1 can hydrolysis ATP, but not synthesis. We have to synthesize ATP, both subunit 0 and 1 subunit of ATP synthase must be work together. And when look at the structure of ATP synthase, ATP synthase can be thought of as a complex of two motors, as I told you, the ATP driven F1 motor and the proton driven F0 motor that rotate in opposite direction. Also, this molecule ATP synthase subunit F0 part consists of three different protein molecules: subunit A, B, and C. When hydrogen ions, protons, flow through the membrane via C subunit, and subunit is compelled to rotate. And we don't know a full structure of A subunit or C subunit, but we know that there is an access channel for protons on a subunit ring in the membrane spaces. And the proton flowing in out from interface the ring C ring system and the A subunit. And in this case, when we look at what drives the specific direction of this rotation, it is always a subunit counterclockwise rotation. Proton loads from high concentrated inner membrane site on the C subunit and release these protons in matrix. And the gamma subunit in the F1 part is fixed to this C subunit of F0 and therefore rotates with it. The alpha and beta subunit in the F1 part, however, cannot rotate because they are locked in a fixed position by the beta subunit, which is anchored to subunit A in the membrane. And we know that C subunit of F0 has at least 10 subunits and bind electrons, bind protons, intercellular matrix, and give the electrons A subunit, and this protons goes to enter to matrix. C ring should be able to rotate in either direction. Ring it cannot be rotated back in the opposite direction because of it is energetically unfavorable. Proton gradient that favors binding protons on the intramembrane spaces and goes to matrix. The proton motive force is thought to drive rotation of a C ring by a transport mechanism using half-channel system. And also C ring can be accepted and half-channel take protons, and protons bounce to this C ring F0 part of ATP synthesis and goes to A, and A leads to protons to matrix. And another part of ATP synthesis in subunit and called F1, the ATP driven F1 motor. How it is work, this F1 subunit, and we have one binding change mechanism for F1 ATP synthesis. The gamma subunit directly contains all three beta subunits. However, each of these interactions are giving rise to three different beta subunit conformation. We know that the ATP binding affinities to the three beta subunit conformation are defined T, L, and O, and T means tight, L loose, and O open, in which ADP and phosphate bind to the O and L conformation. Also, ATP binds tied to the T conformation. And after that, as the protons flow F0 subunit, also subunit rotates in counterclockwise circle, and such as that with each 120-degree rotation, the beta subunit sequentially undergo a conformation change form OLT, OLT, and repeat this changing, and the binding change mechanism model predicts that one full rotation of the gamma subunit of F1 should generate 3 ATP, since each beta subunit have cycled once through the T state. This model, it can be explanation ATP synthesis, even if we don't know everything about ATP synthesis for during oxidative phosphorylation, but we can explain ATP synthesis using this binding model. And when we look at energy balance aerobic respiration, and we know very well glucose has two phases in aerobic and aerobic condition. In aerobic condition, and glycolysis produced 2 NADH and 2 ATP in substrate level, and also this pyruvate dehydrogenase produced 1 NADH. What this reaction turned to 3 times because 6 carbon sugar break down 3 carbon molecules, and after that in citric acid cycle we can produce 3 NADH and 1 NADH, 1 GTP, and 2 times circulation. Totally we produce 6 NADH and 2 FADH2 and 2 GTP. After that, this calculation summary, in aerobic glycosis we can produce 38 ATP per glucose. But sometimes you can read or make different type calculation, such as after that all this reaction we can produce 28 ATP. Also it can be correct because depend on calculation, and in this case second calculation depend on 1 NADH electrons enter the electron transport chain in oxidative phosphorylation can produce 3.5 ATP, and FADH also produce 1.5 ATP, and totally we can calculate 28 ATP. But generally we accepted that NADH electrons, 1 NADH molecules produce 3 ATP, and 1 FADH electrons after entering electron transport chain, and then oxidative phosphorylation produce 2 ATP. On calculation depend on textbook or explanation. But when we look at the energy yield of beta oxidation of palmitic acid, in all of this reaction we can produce 8 acetyl-CoA, 7 FADH2 and 7 NADH, and after that all of these reactions that citric acid cycle, because this acetyl-CoA must be enter citric acid cycle, and totally we produced 128 ATP. And when we look at energy yields by palmitic acid oxidation, totally it is equal 940 calorie, but and it is equal to total standard energy of palmitic acid of 20% of standard free energy of palmitic acid. And we know that this energy of palmitic acid chemical bonds can be used mostly head generation. Also, it is this rule is also we can use this calculation for glucose metabolism. We know that this ATP after that electron transport chain and oxidative phosphorylation produce ATP, and mitochondria, as I told you, have different very important roles in the matrix and also membrane system and electron transport chain on oxidative phosphorylation and ATP synthesis. But it has another most important roles produce or regulation had in our body. It is another important role for mitochondria. And after produced ATP in matrix, we have to move this ATP to cytosol because we know that all derivative way carbohydrate and lipid metabolism occur in matrix and produce ATP, and but all synthetic way in cytosol. For this reason we need this ATP for synthetic way in chemical reactions or enzymatic reactions, and must be transferred this ATP to cytosol, and we used some membrane transport system, And the first one is adenine nucleotide translocase. These proteins can transfer ATP to cytosol and ADP enter to matrix, because ATP also substrate for ATP synthase. For this reason, these two molecules move opposite position and called antiporter system. Another transfer system is phosphate translocase, and the phosphate another substrate for ATP synthase and must be transferred from matrix to from cytosol to matrix. And in this protein system, phosphate translocase is able to do these reactions. And in this case, also the system can be transfer proton from cytosol to matrix and called symporter. Also, we have antiporter and symporter systems in mitochondrial membrane. On the other hand, we can accept an ATP synthase and a channel system. No F0 systems as use transfers protons from intramembrane spaces to matrix. Also complex 1 and complex 3 and 4 are able to transfer protons from matrix to intramembrane spaces. It is two membrane transport system have a critical role for enzymatic reaction in cytosol. When we look at inhibition of oxidative phosphorylation, we have different type inhibitors and affects to activity of electron transport chain and oxidative phosphorylation proteins. And first is retinone. Is this molecule inhibits complex 1, and amital and antimycin A. Also amital inhibits complex 1, and antimycin A inhibits complex 3, and also cyanide can be inhibit complex 4, carbon monoxide, and also sodium azide also can be inhibit complex 4. Especially inhibition of complex 4 more than important clinical site, because especially carbon monoxide toxicity is very important problems for in clinical stage. And another inhibitors, dinitrophenol, is called uncoupling agent, as works in transmembrane proton channel from protons from intramembrane space to matrix. But for ATP synthesis, this transfer must be made by ATP synthase. But this uncoupling agent inhibits this transfer systems, and protons transfer by the dinitrophenol from intramembrane space to matrix. And also another uncoupling agent, pentachlorophenol, also another transmembrane proton carrier. Also oligomycin inhibits ATP synthase, another important inhibitors in oxidative phosphorylation. Malonate is compact inhibitor of complex 2, and atoxide inhibits oxidative phosphorylation by inhibit the transporter of ADP into and ATP out of the mitochondria. It's very important inhibitors for transporter system. Another uncoupling protein is a physiological uncoupler named thermogenin, found in brown adipose tissue that functions to generate body heat, particularly for the newborn and during hibernation in animals. And another important topics in mitochondria Genetics, I want to give some information, genetic structure of mitochondria and mitochondrial disease. And the mitochondrial gene is circular form and does not show a folded structure and no intron structure. And generally, we accepted that and said is a genetic code is universal. But when we look at mitochondrial code, mitochondrial DNA, we will see that the knowledge is not true, because in UGA, in universal code, in mitochondria belongs to tryptophan. Also AGA and AGG in universal code describe arginine amino acid, but in mitochondria explain the stop codon. And AUA in universal code is allosine, but in mitochondria same code is belongs to methionine. Also, we generally also saying that is genetic code is universal. If we have to take one parenthesis, it can be true. And mitochondrial DNA, no addition are made to the cap and poly A of mitochondrial mRNA, also another big problem for mitochondrial genome. Also some, as I told you, some mitochondrial genome codes have different meanings from nuclear codon. When we look at mitochondrial disease, we know very well mitochondrial dysfunction are known to be responsible for a number of heterogeneous clinical presentation with multisystemic involvement. Is mitochondrial disease including multi-system problems, impaired oxidative phosphorylation leading to decrease in cellular energy ATP production is the most important cause underlying these disorders. The main problem is we have some problem in the electron transport chain or ATP synthesis protein. And genetic complexity together with ever-widening clinical spectrum associated with mitochondrial dysfunction pose a major challenge in diagnosis and treatment. An effective therapy has remained elusive till date and mostly efficient in relieving symptoms. When you look at mitochondrial disease symptoms, we can see all important organs affects these problems because of cellular decrease in cellular ATP production. And the heart, skeletal muscle, central nervous system, kidney highly depend on mitochondrial energy production. And major clinical picture or signs are hypotonia and developmental delay, mental retardation, and convulsion attacks and ataxia. Also, increased lactic acid level in body fluid is the most important laboratory finding. Is mitochondrial disease myth, or we describe as a syndrome, has different type sign in the patients. Also, these tissues are more affected by mitochondrial mutation, such as point mutation, deletion, or duplication. When we look at mitochondrial disease, it can be divided into 4 groups. One is missense mutation, protein synthesis mutation, insertion and deletion mutations, and copy number mutation. Also, I want to give information about these problems, mitochondrial genetic problems. First is Leber's hereditary optic neuropathy. With these problems with maternal transmission met as young man, and selective loss of retinal ganglion cells and central vision loss and at end of last stage, we can see optic atrophy. And we know very well where it is problems in DNA of mitochondria. Mitochondrial point mutations are in complex 1, NAD dehydrogenase complex 1 subunit 4, 1 and 6. Also, sometimes we can match some problems in complex 3 and 4. And this Leber's hereditary optic neuropathy accepted that it is rare disease class. Also, maternally inherited progressive disease resulting in irreversible loss of visual acuity and blindness. And second one is neurogenic muscle weakness, ataxia, and retinitis pigmentosa. And we can see some pictures in this disease. For example, neurogenic muscle weakness and dementia, ataxia, and migraine type problems, myopathy, retinitis pigmentosa, and mental retardation. And when we look at diagnosis, multiple biopsy can be normal, and we met some biochemical findings such as abnormal ATPase activity. And we know very well its problem is ATP synthase gene, and we know mutation where it is, and also we can explain some genetic problems and mutation in this disease. And third one, myoclonic epilepsy with red fibers. Problem is tRNA gene has a G change and position 8344, and patients had some problems. For example, muscle weakness, lactic acidosis, and epilepsy, ataxia, also respiratory dysfunction, and also has some cardiac problems and neuropathy. And we can know problems of nerve and problems related to tRNA in mitochondria, in also lysine amino acid. And after that, I would like to give some information between some disease and cancer relationship, because when you look at mitochondria and cancer relationship, we know very well mutation in mitochondrial enzyme generate oncoproteins that result in tumor initiation, and oxidative stress and mitochondrial signal can also support tumor initiation. As I told you, and we know very well, is the mitochondria is the most important free radical sources in the cells because of electron transport chain and oxygen molecules, and complex 1 and ubiquinone complex 3 especially are responsible for production of oxygen-based radicals, oxygen radicals and hydroxyl radicals. And mitochondrial metabolic reprogramming and oxidative stress and signaling can promote tumor growth and survival. Mitochondria additionally regulate redox homeostasis and susceptibility to cell death via alteration of morphology to promote cell survival. We know very well, although we have a lot of finding about relationship between mitochondria and cancer, and mitochondria can be responsible for initiation, growth, and metastasis of cancer processes. And when we look at free radicals in mitochondria, free radicals result to oxidative damage and genomic instability, and after that it can be responsible for cancer invasion and metastasis and angiogenesis. Also, cancer cells change or altered its metabolism in cancer cells, fatty acid synthase, nucleic acid synthesis, and glutamate metabolism and 1-carbon metabolism increases. These all metabolic processes must be increased in cancer cells, especially in nucleic acid synthesis increase because the cancer cells needs a huge amount of nucleotide and nucleic acid, and accumulation in radicals, free radicals, oxygen-based radicals in mitochondria can be responsible for cancer initiation and progression and metastasis. And a second important topics related to mitochondria, apoptosis. We know that is two type apoptosis described. First one is that receptor pathway, extrinsic pathway is depend on that receptor system, and receptor system activated and results to procaspase system, and after that activate procaspase 3 and caspase 3 and goes to apoptosis. And the second one is intrinsic way or apoptosis processes, and it depends on directly to mitochondria. There is some problems in mitochondria, and in mitochondrial membrane, cytochrome C, a part of electron transport chain complex, leads to a matrix of mitochondria and make a complex with alpha proteins, and caspase proteins also make a complex and build an apoptosis. This apoptosis is activated to caspase 9, and end of all processes cell goes to apoptosis. And another important things, aging. We know very well increase mitochondrial DNA mutation, also increase free radical generation in mitochondria, and enhance oxidative damage, also reduce ATP levels and synthesis, and all of them result to mitochondrial damage and dysfunction. All of these problems can be result to aging, and we know very well. Also, we have a lot of publication on aging and mitochondria. After that, all of this information, I want to complete my lecture. I hope to meet another lecture next time. Have a nice time all. Bye bye.